Hello, my name is Marom Bikson, and I'll be talking to you about higher resolution modeling of transcutaneous electrical stimulation, and specifically focusing on how animal models and computational models of animal models can be used to enhance the rigor and the insight of an area which I think is rapidly developing in many exciting directions, including towards treatments for spinal cord injury, more generally, neurorehabilitation, pain, and other indications. But I'm really going to be focusing on the technology that's driving these computational models in this talk today. These are my disclosures, and all these slides can be found online at my Twitter account, as well as a link to all the references I'll be discussing. So I think it's useful to start with a reminder of what dose even means in the context of neuromodulation, including transcutaneous spinal cord stimulation, but really all forms of neuromodulation. And this is something that myself and Peter Schiff and other colleagues spent a significant amount of time trying to define precisely. And this is a summary of the definition that we came up with. But in a nutshell, what we said is that the dose of a neuromodulation device is everything about it that affects the induction of current inside the body. And it is that current then that has physiological actions. So for electrical stimulation, these would be things like electrode size, electrode placement, the waveform that is applied to an electrode, or in the case of multiple electrodes, to different electrodes, the intensity, and so on. So this is the dose, and this is really what we must control in order to do reproducible neuromodulation research and treatment. Now, what do computational models of current flow do? They relate the dose of stimulation, which is something that is outside, with the resulting current flow inside the body. So they relate dose on the outside with resulting current flow on the inside. And it turns out that this doesn't just depend on the electrode placement and size and current and so on. This also depends on the anatomy. So there's the combination of the electrode placement and the overall dose features and the specific anatomy, the shape of different tissue compartments, their resistance, that determines how current flows through the body. And so ourselves and others have specifically modeled transcutaneous direct current stimulation, or more generally, transcutaneous electrical current stimulation. And a lot of this work has derived from our prior work modeling transcranial direct current stimulation. So these are models here specifically of transcutaneous spinal direct current stimulation. And these are just some exemplary montages. You can see in one case, this is a MRI-derived model. It's of myself. In one case, you can see there's an electrode on the cheek with a return on the shoulder. Another case, an electrode on the back of the neck. And in the third case, actually an electrode on the top of the head with a return on the shoulder. So that's driving current all the way down the axis. And in each case, the computational models will predict how much current gets into the brain, into the spinal cord, where it travels, the intensity in different regions. And the approach that my group has taken and others to transcranial spinal direct current stimulation really derives from our work with transcranial direct current stimulation, where the emphasis is on developing models that capture the right kind of details, the right kind of precision. So that means representing the most important tissues and assigning them the correct properties. And in the case of transcranial direct current stimulation, we focused on the cranium, on the cortex. But in the case of transcutaneous spinal cord direct current stimulation, there's now this importance to develop very accurate models of the spinal cord itself, and equally important, the surrounding tissue. So the vertebrae, other soft tissue, muscles, skin, all the way out to the surface. But today I'm not going to be focusing on models of current flow in humans, but the work we've done to develop these models in animals. I should say that it is now very well understood that the same dose applied to different people will produce a different current flow pattern through the body. And that's expected. If the current flow pattern depends on the anatomy, then different anatomies will produce different current flows. And this effect obviously is amplified when you're talking about extremely different anatomies, such as when going from humans to large animals and then to small animals. And this brings up a very important point. It suggests that if we want to do translationally meaningful animal studies, and by that I mean animal studies where the target tissue in the animal received the same electromagnetic fields as would be produced in a person, then we can't give the animals the same dose as the people. Giving 1 milliamp to a person is not the same thing as giving 1 milliamp to a mouse. Instead, we need to adjust the current that we give the animals, typically reduce them because they're smaller, so that the electric field, the current density produced in the target tissue, is the same between animals. And I'll be talking more about that. Let me go back to the example of transcranial direct current stimulation to show how we've applied this kind of thinking there. This is specifically in the context of developing safety standards. So safety trials of TDCS have been done often in rodents, and here the current is applied transcranially, often using epicranial electrodes to the rodent brain, and the current is increased to very high levels until an injury is produced. And this in turn is used to establish safety thresholds in people. And here's just two examples of two different TDCS approaches that were used in two different labs collecting safety data. Now it's both in rats, but you can see that the electrode here placed over the head is a slightly different size. In panel D, it is larger than it is in panel B, but you'll also notice the position of the return electrode. The counter electrode is different between these two cases, and they use comparatively similar currents. However, the resulting electric field intensity in the brain was very different, as was the pattern. And so the point here is that there's a lot of nuance even when you're talking about specific animal trials and comparing within animal trials, never mind comparing to people. So here you see 500, about 0.5 milliamp applied to rodents, and here's 2 milliamp applied to a human, in this case me again, and these are on the same scales. And so as could be expected, less than a milliamp, a fraction of a milliamp applied to an animal produces much higher electric fields in the brain than several milliamps applied in a person. This emphasizes this notion of scaling and how we need to think about scaling when going from animals to people, but equally thinking about specifically how we dose in the animals and how small differences in dose in animals can also have this impact. So talking about now transcutaneous spinal direct current stimulation in animals, we've been doing this work now for about half a decade, initially working with rodent animal models and collaborating with individuals like Jack Martin and his team at City College, where they're trying to develop neurorehabilitation approaches using the stimulation technology. And we were supporting that translational effort by developing these computational models. And as we've done when we've developed computational models in other domains, we really tried to focus on creating models that captured the right kind of detail. And these are image-derived models, and so they're very much dependent on the ability to collect very high-resolution images. In this case, the rodent scans were based on 7T scans, as well as CT scans. So the MRI scans giving us contrast for soft tissue, and the CT giving us exquisite contrast when it comes to bone. And so these two kind of scans were co-registered to create the single model that you're seeing here. And so we simulated the current flow in these cases. And what you're showing here are very small variations in montage. There's actually 3 variations, and you can see that both electrodes are being moved around. And what you're seeing is that if you move a single electrode or both electrodes, the distribution of current flow pattern changes. And one of the points I want to emphasize here is that often people focus just on the electrode that they would consider the active electrode, the electrode that they consider to be more important. But as with other applications of neuromodulation, where you put the counter electrode, what you're calling the return electrode, will equally be important in influencing overall current flow pattern. The other thing I want to show you that you can see here is as we move these electrodes, and they're still positioned broadly over the spinal cord, we start to get more and more stimulation into the cortex, into the cerebellum itself. And so this is also an important factor to consider. These animals are much smaller, and it's possible that a montage in an animal that one might hope restricts current to just the spinal cord would spread to other parts of the central nervous system, which may complicate the interpretation of any findings and may also compromise the ability to translate any findings to people. And again, one of the essential things we're trying to do with these animal models is relate intensity. So consider how, for example, in this case, 1 milliamp applied to the animal produces a current density that is significantly higher actually than what you would expect in people. This does not in itself invalidate the work. In many cases, when you're doing work with animals, you're looking for big effect sizes. You're looking to see very categorical changes. So in some cases, this actually makes a lot of sense. Even so, it's very important to be cognizant of the differences in dosimetry between animals and the human case. The other thing we notice when we develop these very detailed models is what you're seeing here, where you're seeing these current concentrations that are, you can almost see little stripes of current concentration, and it's clear that the current is sort of being funneled between the bones. And so where you have these gaps, you have higher concentrations of current flow. But that also tells us that therefore it must be really, really important to simulate bone correctly, as well as other soft tissues, right? So the fact that we're seeing these idiosyncrasies in current concentration because of the surrounding tissue also tells us how important it is to precisely represent the surrounding tissue. Going back to the notion of really focusing on generating models that represent the right kind of detail. Also working with Jack Martin's group, we've extended these models to consider injury. And again, when it comes to the, in this case, the neurorehabilitation applications, I'll refer you to the original paper. Here, I really just want to focus on the current flow modeling itself. So in these cases, the spinal cord has been injured, as well as potentially some of the bone around it. And so that is represented in these models where we can scan these animals that have had an injury and ask the question, now when we apply transcutaneous spinal direct current stimulation to them, how is the pattern of current flow altered? And generally speaking, the lesioned area, which had relatively resistive gray and white matter in it, has now been cannibalized and it's been replaced in the chronic stage by much more conductive cerebral spinal fluid. So that cerebral spinal fluid we found acts to funnel the current towards it. This is something that's found also in other models of injury. Now, to the extent that that area that has been cannibalized doesn't have any brain left, we're not so interested in the fact that current has been concentrated there, but we also find that current can be concentrated in the penumbrum, or the region just around that lesion. And that is very interesting because that is often the region where one is hoping to produce neuroplasticity. So the fact that more current can be driven there by the presence of the injury is an interesting finding that has translational relevance. And so I started with this big picture idea that we shouldn't give animals the same dose that we're giving people, that we should probably be reducing the dose, at least in those studies where we really want to be comparing apples and apples, where we want to be applying at the target tissue in an animal the same intensity that will be provided at the target tissue in the people. And so that's why we may want to adjust the current appropriately. How would we do that? Here, really computational models are playing an essential role. And generally speaking, you can think of it as a two-step process. So the first thing you do is you predict the electric field in the target in the clinical case. So if you're doing transcutaneous spinal direct current stimulation, you would position the electrodes where you might think to be applying them in the clinical case. You would predict current flow through the spinal cord, and then you would pick a particular ROI with a particular electric field, because you can see that the electric field is not uniform everywhere. So there is no one electric field. You have to select a region that you think is important for your particular outcome, and you extract the electric field there. Now, the next step is you run a comparable model in an animal. And here again, you could have a large animal, small animal, and you to represent the animal as well as the electrode montage and placement that you're going to be using in the animals. And in the animal model, you predict an electric field in a comparable ROI. And what you do is you adjust the current that you apply in the animal model such that the electric field in the animal ROI is the same as the ROI in the person. However, and this in principle is the approach that's done, but there are several issues. The first one I already alluded to, that you have to pick an ROI. But in many cases, there may be more than one ROI. It may be very hard to say that just a single region of the spinal cord is responsible for all the outcomes that you see, and no other region is responsible. So you might be thinking of multiple ROIs. But this creates a problem because the spatial distribution of current flow in, let's say, a human case is nothing like that in an animal case or in the rodent case. So if you want to match the electric field in one area, you know what I mean, let's say in one segment, and match that in the animal, you can't necessarily do that for another segment or in another segment in another segment. The spatial extent, the relative spatial extent of how current changes across the animal is not going to be the same. And so even though you match the electric field in one ROI, you're mismatching it in most of the other places. And there's really no apparent way about that. And there's even a deeper problem, which is if you zoom into any one of these ROIs, gray matter, white matter, a root, whatever it is, and you look at it closely, what you'll see is that there isn't just one electric field. The electric field is not homogeneous, it's not uniform. And so therefore it's changing from one side of your ROI to the other side of your ROI. So then the question is, well, then what is the electric field in the ROI? There isn't just one, and there isn't just one in the animal, and it's impossible to precisely control these or precisely reproduce them. So there seems to be some problems with this very simple 1-2 kind of approach. Nonetheless, it's done. It's done because you have to do it. And so the framework that we propose to explain this approach is something that we call the quasi-uniform assumption. And it sort of follows the same rule with a little caveat that one, you predict the electric field in a region of interest, and you assume that it is quasi-uniform, that is essentially uniform. What that means is you assume, you make an assumption that in the ROI that you're interested in the clinical case, there is a single electric field that can be extracted. We now run the simulation in the animal, and we once again assume that even though the electric field may not truly be uniform across the comparable region of interest in the animal, we assume that it's quasi-uniform. And so we're taking one electric field and matching the electric field again under this notion. And the bigger issue, which is that the electric field can't be matched everywhere, is still overriding. But with the quasi-uniform assumption, we sort of accept it. We just say we can't match the electric fields everywhere, so we're only going to do it in the ROI. And though we gave this thing a name, it's actually done universally. It's just done implicitly, because otherwise there's really no other way. It's done implicitly when people are doing these animal models and they're trying to scale things appropriately. It's actually done in the computational models themselves. When people are running computational models and they're reporting an electric field here and they're reporting an electric field there, just the fact that they're assigning an electric field to a region means that they're saying, well, there's a quasi-uniform, there is a single value of electric field that we can read from these regions. So when you're seeing a prediction of current density or electric field across an animal or person, there's a quasi-uniform assumption caked into it. I want to now say a little bit more about the work we've started to do using larger animal models, and this work is ongoing. So just to be giving a general overview of it. Once again, as we're moving to a new animal species and a new, and in this case, interested in the spinal cord in this animal species, our attention really starts with how we're going to actually obtain the images that we will then use to segment the tissue in order to generate the models. And so there's a lot of piloting that's done, looking at different imaging sequences, looking at constraints, such as the condition the animal can be in during the scan, depending on how long the scan is. Whereas, for example, for a smaller animal, we may be able to use a small bore CT. With a larger animal, we may not be able to use it, or we may need to do multiple scans. We're slowly moving the animal through that bore and afterwards process these images together. So in short, a lot of thought that goes into how we collect these images specifically for the purpose of generating models, right? So this is thinking about a pipeline already. So these are the kind of models that we are generating for cats. You can see the attention to the segmentation, and grossly speaking, certainly this is a step towards the human situation as far as overall scale and morphology. In this case, gray and white matter are clearly distinguishable and can be separated out, so we can create these models of cats, and we can simulate stimulation being applied with them. This again is in collaboration with Dr. Martin's group. They have been working with these animals as a model for neurorehabilitation. So cats that have a spinal cord injury, and then looking at whether this form of stimulation can support healing in these animals. And again, here I'm just going to be focusing on the computational modeling and imaging component of it. I should say, though, that right away we're seeing that it's much easier to draw analogies between the kind of current flow distributions that you're seeing in the cat and the human than necessarily what we would see in the rodent. If you saw earlier some of the models I showed for people, there was a lot of concentration, particularly around the roots. Those are the kind of features that we saw to some extent in rodents as well. We're seeing very nice homologs to that when we're simulating what's going on with cats. The other thing we started to do is like these these animals are much more precious, and the modeling pipeline is much more complex. And so what we're starting to do is, for example, when we're considering injury, rather than necessarily imaging a series of injured animals, we are rendering an injury. So we would start a situation where the animal is a healthy animal with all the anatomy intact. We would scan it, and then we would synthetically create specific injuries by removing bone, by removing gray and white matter in a way that we think is consistent with how the injury is being created in the animal model. Here you see an example of a simulation like that, where the blue trace is the standard anatomy. The electrodes here, you can probably guess, are roughly positioned over where you're seeing the electric field increase. You can see that sort of oscillating electric field peak intensity that's defined by the anatomy and specifically the bone structure around the spinal cord. In the case of the injured animal, what you're seeing here is that some of that tissue has been removed, and similar to what we saw in a rat, but in a much more amplified way, we're seeing current concentrate around the injury, not just in the injury itself, but in the space around the injury. And so while we saw this in the rodent, it is much more dramatic when we're going to the scale of the cat. And this goes back to, and this has, I think, important implications to what we think would happen in people, and more general implications to this notion that, for many reasons, using larger animals can be a powerful tool to develop translational treatments. But here I'm showing you specifically, they may be a more powerful tool to develop neurorehabilitation treatments, because the current flow patterns that we get may start to be more analogous to what we see in people. So that was a very broad overview, but I hope it illustrated where we're placing importance in the developing of these models, specifically from a methodological perspective. As I mentioned, All these slides and references are online, and in many of these papers, there is actually there's an analogous neurophysiological behavioral study happening in these animal models that we're using to validate them. But that was not my focus for today. Thank you very much for your attention.